Hello. First, I would like to thank the organizer for allowing me to present my work today. My name is Lindsay Goulet from Metagenomics unit at INRAE in France. Today, I'm going to talk about Crocodile, a deep learning tool for the detection of well-to-well contamination in metagenomic data. So metagenomic sequencing allows a deep characterization of microbial communities without prior organism isolation or culture. In practice, the first step is sample collection, followed by DNA extraction, then sequencing, and finally mapping to obtain the species abundance table for each sample. From collection to sequencing, there are multiple biases, such as contamination. We can define contamination like the presence of DNA that not come from the biological sample under study. There are two types of contamination. The first type is external contamination from an environmental source or lab reagent, for example. The second type of contamination, which will be the subject of this talk, is contamination from one sample to another, processed on the same plate. This is what we call well-to-well contamination or cross-sample contamination. Currently, well-to-well contamination can lead to biased results, such as overestimation of alpha diversity or false strain sharing events, and eventually to false conclusions. So the detection of this contamination is crucial, but the question today is how to spot it. Currently, there are two methods used for the detection of this contamination. The first uses negative controls on the plates, but this method cannot be used on all data already generated, and the negative control are costly. And also, this method don't discriminate contamination sources from contaminated samples. The second method uses strength results analysis, but this method don't work with related samples like mother and child, and also not discriminates sources from contaminated samples. This is why today we introduce a new method, Crocodile, for the detection of this contamination. This method is based on abundance profiles. So first I will define what an abundance profile is. So we start with the abundance table of two samples here, and we plot the abundance of species in sample 1 here in function of species in sample 2 here. So each point in pink corresponds to a species, and species on the edges of the plot are those present in only one sample. So the species here are only in sample 2, and species here only in sample 1. The comparison of species abundance profiles reveals specific patterns associated with well-to-well contamination. Indeed, above a certain threshold, all the abundant species on the contamination source sample are present in the contaminated sample. Thus, species that were not present before the contamination event, in yellow here, have a proportional abundance between the two samples and form what we call a contamination line here in purple. In addition, we can estimate the contamination rate from the value of the intercept here in orange. But comparing samples 2 by 2 represents a significant number of plots to inspect. For example, for a 96 well plate, this represents 4,560 profiles to check. So we have therefore chosen to restrict ourselves to sample pairs with a high correlation because the contamination line increases the correlation between samples. But correlation like sensitivity or specificity for certain kind of cohorts, like samples with a high or low correlation. Indeed, we can see on the left here that samples are not contaminated because there is no pattern of contamination, but they have a high correlation. And on the contrary, on the right, samples are contaminated because we can clearly see a contamination line, but they have a low correlation. This is why we developed Crocodile, a deep learning tool for the detection of well-to-well contamination. So Crocodile is a dense neural network and takes as input the most abundant species in the potential source sample and outputs the class prediction, either contaminated or non-contaminated. This is a representation of the model of Crocodile. For the training step, a semi-simulated contaminated dataset using real data and varying the contamination rate and sequencing depth was generated. For the non-contaminated dataset, one part uses real data with simulated different sequencing depths, and the other part uses sample pairs from different cohorts. These samples are not contaminated because they have never been processed together. Then we split the data to training and test datasets. So we have 80% for the training and 20% for the test. Performance was evaluated on 3 real and curated datasets. The idea is to have a method that can be used for any type of sample without having to modify the threshold according to the correlation between samples. So this is a ROC curve, and the points on the curves corresponds to the thresholds used for the clash prediction. So we have 0.6 for the correlation method and 0.5 for crocodile. First, we can confirm that the correlation method don't work for the low and highly correlated sample, like I said before. And next, we can clearly see that crocodile outperformed this method for the 3 datasets. So to sum up, we have introduced today a new method and tools, crocodile, for the detection of the well-to-well contamination. We have seen that crocodile outperforms the correlation-based method, but there are still many false positives without a contamination line. This means that the model doesn't use the same criteria as human eye. This is why, at this time, we recommend crocodile as a net for human inspection in order to reduce the number of plots to manually inspect, rather than a fully autonomous approach. Next, we intend to improve the training datasets to increase model performance, and crocodile will be used to perform quality control of public cohorts, and the result will be integrated into the MIAS database. If you don't know what is the MIAS database, you can go to the poster B167. And finally, we will extend the use of this tool to identify contamination rates and working towards the automatic decontamination of samples. If you have any question, I will be happy to answer them at the poster session at 6 p.m. Or feel free to email me. And I would like to thank all the people who helped me and work with me on this project, especially Guillaume, who are here today. And thank you all for listening to me.